Okay, so by now, if I have the timing right, you have learned all about signaling cascades. You've learned about the ligand binding to the receptor. You've learned about the signaling cascade molecules that transfer that signal and then lead to a response. And you've even learned about different types of signaling and threshold and all these key things that we've learned in Chapter 8. So what I want to do just at the end is walk through two key signaling pathways that you need to know fairly well. And the big part of this is really just starting to recognize what's a ligand, what's a receptor, what's a signaling molecule, and so forth. But I also am going to spend a little bit more time on the G-protein signaling pathway because this is such a common signaling pathway that it's one of the ones that you really do need to understand pretty in depth. All right, so overall goals, if you're given a pathway, maybe even one you've never seen before, you should now be able to identify what's the ligand, what's the receptor, what are the signaling cascade molecules, and what's the cellular response. And ultimately, you should be able to also, by memory, kind of describe the first steps of one particular pathway, and that's the G-protein pathway. All right, so this is the EGF pathway. This is the epidermal growth factor pathway, and this actually is the pathway that causes one cell to signal other cells to start dividing. So this is a paracrine signal. The ligand is secreted, and then it's going to bind to receptors and ultimately lead to gene expression changes that causes the cell to divide. And so we can see this here if we start focusing in. We have the epidermal growth factor. This is the ligand here. And this ligand forms a dimer, right? So it's two EGF proteins together. This is quaternary structure. And this is important because when the dimer EGF binds to the receptor, it actually pulls two receptors together. And this is absolutely critical because normally these receptors here, this transmembrane domain, this purple thing is the receptor, right? It has an extracellular domain that can bind to the ligand and an intracellular domain that leads to a change that the cell can basically adapt to or sense inside that cell that indicates a ligand has bound outside the cell. So again, key concepts with the ligand binding to the receptor. The ligand, when it binds to the receptor, it needs to cause a change that is sensed on the inside of the cell. So the inside of the cell, those signaling molecules know that a ligand is indeed bound. So normally these single transmembrane receptors are floating through the membrane. So remember, our membrane flows laterally. And so they're not together normally, but if the ligand binds, it's going to hold one receptor, and then it'll eventually bind to the other receptor, and now that ligand is holding these two receptors together near each other in the membrane. And so, basically, when the ligand binds and dimerizes the receptor, brings two of them together, those two receptors now, being in close proximity for a long time inside the cell, they can actually cause phosphorylation of each other. And remember, we talked about phosphorylation. This is basically turning on a protein that was previously off. So when those receptors are just flowing through the membrane by themselves because there's no ligand bound, there's nothing to phosphorylate each other. They're off. But as soon as they're held together, this one phosphorylates that guy, and this guy phosphorylates that guy, thus turning them on. So now we have a change inside the cell that is unique and only happens when the ligand is bound. So this phosphorylation then is now going to turn on those receptors, and those receptors now are able to bind to our first signal cascade molecule called GNRP. And then GNRP is active. It's actually going to be a kinase, and it adds our third phosphate to GTP. GTP is just like ATP. TP is triphosphate. So now we have three phosphates on this guanosine. And this is a molecule bound to a protein called RAS. So RAS, when it's bound by GTP, is on. RAS, when it's bound by GDP, is off. So the activation of our receptors by phosphorylation, which brings GNRP, then causes RAS GDP, which was off, to be turned on, now RAS GTP. This RAS GTP, now that it's on, can go on and keep activating our signaling cascade. It's going to turn on a protein called RAF, which turns on MEK, M-E-K. K stands for kinase. Kinase is going to phosphorylate ERK, also a kinase, and then ERK can activate our transcription factor, and this now leads to turning on new genes that were previously off. And these genes in this case would be genes that basically help the cell divide because it was just signaled by EGF that it should divide. That's our signaling cascade, and now we have genes that are on that were off before, and it's all based on the ligand binding to the receptor, leading to this cascade of events where all of these molecules, GNRP, RAS, GTP, RAF, MEK, ERK, all of those get turned on in sequence to lead to this cellular response. Now, we do not want this to stay on forever. That would be bad. That would actually cause cancer, because if once a cell was told to divide once, it kept dividing forevermore, that's cancer. So we want this to be turned back off by default, and so this is actually the role of GAP. GAP's job is to take RAS GTP and turn it back to GDP. And this is actually recruited by our receptor as well. So basically, this is kind of an auto negative feedback loop. So the same thing that activates this whole signaling cascade to cause cell division is actually going to activate the thing that turns it off as well. And so what GAP does is it turns RAS GTP back to GDP, thus turning this whole thing off. The rest of the signaling cascade would stop. And so basically what that means is this is only allowed to be on for a certain amount of time in the cell. That would be long enough for the cell to divide, and then it's going to automatically be turned back off, and unless the ligand continues to be there turning this on some more, then the default pathway is for the cell not to divide. And now it's turned back off, and the only way it'll divide again is if a little bit later we get a new ligand telling it to divide, because maybe it's time for it to divide again. Alright, the other pathway I'm going to spend some time on is the G-protein coupled receptor pathway. And this is the one that's very common, and so at least the first few steps are similar between a lot of different cellular pathways. So it's an important one to know because it's an important one for cellular function and for life. I also have a review video on this. It's about 3.5 minutes, so you can watch this video now, and then when you want to review this and kind of remind yourself how this G-protein coupled receptor pathway works so that you can fully understand it for the exam and for your own knowledge later on, you can review it in that later video. Alright, so let's talk about it here for a second. So again, we have a ligand. This is our G-protein, and then ligand, and then the G-protein coupled receptor. So this is the G-protein here, this is the ligand, this is the receptor. And you can see we have a default pathway. When the ligand is not bound to the receptor, it's at a certain conformation that doesn't allow it to bind to the G-protein. Now here's the first key with this, right? The G-protein is initially off, and this G-protein has 3 subunits, the alpha, the beta, and the gamma subunit. And when the alpha subunit is bound by GDP, guanosine diphosphate, then the alpha subunit and beta and gamma are all together. And when all 3 are together, this is actually off. So this makes sense because our default pathway is off. This signaling molecule is not signaling, it's off because our ligand has not yet bound to the receptor. However, when this ligand does bind to the receptor, that causes a conformation change in the receptor. And now this receptor, the cytoplasmic domain of it, the one part that's inside the cell, that can actually bind to our G-protein. And by binding to the G-protein, the receptor is going to cause GDP to be released from the alpha subunit in exchange for GTP. So take-home message here so far is that when the alpha subunit is bound by GDP, alpha, beta, and gamma are all together, and that's off. When the ligand binds to the receptor now can cause the alpha subunit to bind to GTP instead of GDP. And when that happens, the alpha subunit and the beta gamma subunits separate. So you can actually think of the beta and gamma subunits kind of as an inhibitor of the alpha subunit. When they're together, it holds this off. When they're separate, this alpha subunit, now bound by GTP, is on. And the whole purpose of the ligand binding the receptor was to take the GDP and exchange it for GTP, thus turning this on and causing the beta-gamma inhibitors to be released. So this is how the ligand binds to the receptor and causes the G-protein to turn on. Once the G-protein is on, so that alpha subunit's by itself, no longer bound to the beta-gamma inhibitors, now it's going to start a downstream signaling cascade. So the activated alpha subunit of G protein is now going to bind to this green blob here. This is called adenylyl cyclase. So when the alpha subunit binds to adenylyl cyclase, it turns it on. It turns the next step on. And then adenylyl cyclase, now that it's activated, it's going to take ATP in the cell. And ATP is abundant in the cell because that's our energy, but it also can act as a signaling molecule. And so when ATP is converted to cyclic AMP, right, monophosphate now. So now we've actually taken two of those phosphate groups in our triphosphate and gotten rid of them, and now it's monophosphate in this little cyclical version. That is the on version of this. All right, so remember, cyclic AMP is only made when adenylyl cyclase is on. Adenylyl cyclase is only on when the alpha subunit of the G protein is bound by GTP and therefore active. And that only happens when the ligand binds to the receptor. And so now that we have active cyclic AMP, it's going to bind to this protein here called the pKa inhibitor. Normally, the pKa inhibitor is inhibiting pKa from doing its job downstream. So in the absence of cyclic AMP, the inhibitor is keeping pKa from doing its job. But when cyclic AMP binds to the inhibitor, now the pKa is released, right? So I've blocked the inhibitor. Now pKa is released, and pKa, as a kinase, is able to activate the next protein. And this allows basically the rest of the signaling to occur. We're not going to worry about it from here, but this allows the cell to respond to whatever signal the G protein ligand is telling it to do. Right? So this is the key aspect here. We've made cyclic AMP. Cyclic AMP is needed to block the inhibitor from binding and inhibiting pKa. So only when cyclic AMP is around is pKa active. So basically you can think of cyclic AMP as activating pKa. Now pKa can go downstream and turn on other things in the cell through that kinase cascade that we've learned about before. But again, we want to be able to turn this signal off. So when we have less ligand, all right, so the ligand's running out, the signal should be off, the ligand is going to be released. It's going to go underneath the threshold, and therefore it's not going to bind to the receptor anymore. So now the receptor is back off because we have less ligand. So when that happens, there's nothing activating the alpha subunit, and GTP eventually will turn back into GDP. So that turns itself off, basically. If there's nothing keeping it on, it'll turn itself off. And then that alpha subunit with GDP binds back to beta gamma, and that now turns this off. And so when the G protein is off, it is no longer able to activate the adenylyl cyclase, and now the rest of this is also off. We're no longer turning the rest of this pathway off. There are some enzymes already in there that are going to take the cyclic AMP that was holding the inhibitor, and they're going to get rid of them. It's going to degrade those, and so now if we're not making new cyclic AMP, our inhibitors are active, right? So the pKa regulator that inhibits pKa is back to inhibiting pKa. Now we're no longer activating new signals. And then all of our downstream phosphorylated proteins, they're always going to be dephosphorylated as well because we want them to turn off when there's nothing left to turn them on. So again, the main take-home message here is there are ways the cell can automatically turn a pathway off as soon as the ligand is no longer around. So the ligand binding to the receptor has to keep that pathway on, and the default is for the pathway to turn back off as soon as the ligand's not keeping it on. So the default pathway of a signaling pathway in a cell, the default of any signaling pathway in the cell is to be off because we only want that cell to respond to the signal when the ligand, the signal, is there. Hopefully that helps. Use the review videos and bring questions to class, and we'll work on this together as well. All right, good job.